In this video, we are going to study that how biotechnology is being used in agriculture. The biotechnology in agriculture is being used extensively, and one of the major applications of biotechnology in agriculture are or is the development of pest resistant crops. Now these pest resistant crops actually had the advantage of using less amount of pesticides, or we can say that biotechnology is helping the farmers by the less application of pesticides, which are chemicals. Now here we are going to make use of a bacteria, and this bacteria over here which we are going to use is Bacillus thuringiensis. This bacteria actually has a gene. We call it as Bt gene. The name we get it from here, B and T. So we call it as Bt gene. This Bt gene is responsible for producing a Bt protein. Now this protein is actually being used in plants. This Bt gene is utilized in plants to develop the resistance against pests and insecticides. So it is actually a biopesticide. It is an example of a biopesticide. Here, it has been incorporated into corn, rice, tomato, potato, soya bean, etc. It has been used extensively and therefore is of a great interest. Now, various strains, some of the strains of the bacteria are actually they act on wide variety of insects such as lepidopterans. So it is actually working against many terans. It is acting against that lepidopterans, that is armyworm, tobacco budworm, etc. Then it is also acting against coleopterans. And also diopterans such as mosquitoes and fly. Diopterans, that is mosquitoes and flies. So it's applicable on wide variety of insects as well. And actually it produces protein crystals during a particular phase of their growth. Now these are actually, when we are talking of this Bt gene is toxic gene. These are the species of the insects on which it is infection. That means it destroys these or provides immunity against these to the plants. Then Bt gene, actually when we are talking of this protein, this is toxin. We call it as toxin, Bt toxin protein. Now this is actually this cry gene or cry protein, that is Bt gene will produce the Bt protein. We call this Bt gene as cry gene also, because it synthesizes the protein which is crystalline. This protein is crystal. So therefore the name cry gene. Now this protein is crystalline, and when dissolved in alkaline medium, it turns toxic. So what happens is, this bacteria has the cry gene or the Bt gene, which produces the Bt protein, which is toxin and crystalline in nature, therefore the cry gene name. Now this Bt protein in alkaline medium, it turns toxic. This is the property of this particular protein. Now, it does not affect the bacteria though. Bacteria protects itself because it does not have the alkaline medium. So the bacteria is well protected by the action of this particular protein, since there is no alkaline medium over there. But if alkaline medium is provided, it turns toxic. Now this was isolated, that means this Bt protein was isolated and it was incorporated into plants. It was then isolated and introduced in plant. That means it was through the process of separation and the technique, that is this gene over here, we call it as cry gene. So the cry gene over here is isolated and incorporated in plant. Once it has been incorporated into the plant, the process of translation and transcription happens. Now this translation and transcription causes the synthesis of the Bt protein or the toxin protein inside. Now the plant has protein, toxin protein. Now the thing is, it will also not cause any damage to the plant because plant has no alkaline medium. When the plant is infested with larva, that is the larva, or when the larval stage, when that feeds on the plant, this toxin protein enters into the gut of larva. So what happens over here? Toxin, that is the toxin protein enters into the larva of the gut of the larva, where the medium or the pH is alkaline. So here the pH is alkaline. As a result, this protein turns toxic and kills the insect gut because it causes perforations in the epithelial membrane or epithelial cells of the insect's gut. You have to remember that this Bt protein or Bt toxin exists as inactive protoxin, but in the plant, but when it is taken in by the insect, it is converted into active form of toxin due to the alkaline pH of the gut, and it causes the active toxin will bind to the epithelial cells of the midgut and will cause pores or perforations over there, thereby the breaking or the lysis of the cell and finally causing the death of the insect. So specific Bt toxin genes are isolated and incorporated into several crops. For example, cotton, and cotton is a very good example. Now the genes, the the type of genes which we are choosing depends upon the crop and the pest. Like the type of gene which has been isolated from here has to be effective on certain kinds of pests only. Therefore, we have to choose accordingly. And the Bt toxins, because the Bt toxins are insect group specific, they are not applicable for all the types of insects, but specific type of insect. And the cotton, that is the Cry 1AC and Cry 2AB, control the cotton bollworms. That is, the Cry 1AB controls corn borer. So we have different kinds which are being used in developing pest resistant crops.